So you can see, so we form what? This structure by addition of a proton, and we form this structure by addition of what? A base. So amino acids can act as what? We can add acid to an amino acid to form what? This structure. We can add a base to form this. Okay, so titration curves are obtained when the pH of a given volume of a sample solution varies after successive addition of acid or base. The curves are usually plots of pH against the volume of titrant added, acid or base. So let's look at the titration curve. So with this titration curve, so we have glycine as an amino acid, pKa 1, pKa 2. So meaning that this is the titration curve. So we'll add 0.5 units or 0.5 concentration of what, or units of what, OH or a base, 0.5 equivalent of a base. We add it to this amino acid. So when we add it, this is an acidic amino acid. So the H will react with the OH to form water. So this H will disappear. So we can see negative. So H is not there again. So this one becomes what? Basic. Then pKa2, we can see that this one of these H will disappear. Will react with the OH to form water. So we get NH2. Do you understand?
Please go over again.
Okay, this is we are looking at isoelectric pH or PI or isoelectric point. And I'm saying that we are having a base. This x-axis is what? Base or OH minus equivalent. Then this is a pH. The y-axis is the pH. And you know pH starts from 0 to 14. But we have up to 13 because we have shortened it because of this reaction. So this is an amino acid. This amino acid is called what? What kind of amino acid is this? What type? You know that what is the side chain over there?
The side chain.
Uh-huh.
The side chain is a CH.
Okay, this is the amino group. This is the COH, the carboxyl group, or the carboxylic acid group. You have to identify the constant H and the side chain. So what's the side chain?
The side chain is the CH.
Let's go back. Let's go back. This is the structure. This is glycine. You are saying CH, also CH2. But this H is always constant. You can see the H is always constant. This one too is always constant. This one is constant. All true. COO minus is constant. NH2 is constant. The only thing that changes is what? The side chain. So because you are seeing CH2, one of them should be H, the constant H, and one of them should be the H that constitutes the side chain. Do you understand?
Yes, sir.
Okay. So let's go back. Let's go forward to our slide. So this is the structure. So this is the structure. Let me pause the recording small because of the dog there. So we have, so Annabelle, let me ask Florence. Wait, do you have Florence here? Florence, don't call. Florence, don't call. Florence. Hello. Yeah, some more. Florence.
Hello, so
Omia ne mute in na onkasa. Florence.
yeah.
Florence, what's the name for this amino acid? This one.
Which one?
This one.
I don't be sure. That is the point.
Where the pointer is?
Where the pointer is on top here. Is it on? Yeah, just.
Is the Florence and Tamlenko? Okay, then Felicia. Eh? Yeah, what's it? Me also. Felicia. Then talk. But we are hearing you. Okay, Raphael.
Sir.
If you are not talking, mute your mic. Raphael.
Sir.
What's the name for this amino acid?
Sir.
What is the name for this amino acid? It's written here. Glycine. And you're saying, Hey, Mamo. Clap for yourselves.
No, that we are both of
Yo, so let's go back to our. Okay, so let's continue. Okay, so we are having, so when we add 0.5 equivalent of hydroxyl ions or base to this one, the hydrogen and hydroxyl ion, this hydrogen will react with the OH to produce water. So this hydrogen will disappear. So we can see the hydrogen has disappeared over here and water has been formed. The same way, we add another 1.5 equivalent of the hydroxyl ion. When we add it, one of these hydrogen, this is 3 hydrogens, one will disappear, so I will get 2 hydrogens here. And the one that will disappear will react with this OH to form water. So now we have 2 isoelectric points here. We have 0.5 that we added. So it will generate a pKa of 2.34. The 1.5 equivalent will also generate a pKa of what? 9.6. When you add 9.6 to When you add 9.6 to 2.34, we get what? 5.97. Do you have calculator? Does anybody have calculator?
No, sir.
Okay. So let's go on. So these ones are examples. Go through, you understand them. Three letter abbreviations and one letter symbol for amino acids. So glycine, the 3-letter abbreviation is GLY, and the 1-letter abbreviation is G. What is an abbreviation?
It's a short form of a word.
Okay, so 3-letter short form of glycine is gly. So anywhere you see gly, even on the internet, it is glycine. Ala is alanine. Pro is proline. ASP is what? Aspartate. When you see D, is also aspartate. When you see R, is arginine. When you see H, is histidine. Lysine is K. Leucine is L. Valine is V. So we have the 3-letter notation and 1-letter notation or symbol. The same, so you go and study these ones. I may bring some of them in the exams. So what is an essential amino acid? And what is a conditional amino acid, or conditionally non-essential and non-essential amino acid? An essential amino acid is the amino acid that the body cannot produce. You know, the body produces amino acids. The body produces, like the tyrosine and the rest. The body produces them for various enzymatic reactions and also to form the basic proteins of the body. But if the body cannot produce an amino acid, then that amino acid is called essential. So we have to get it from the diet that we eat or from the food that we eat. That is essential amino acid. A non-essential amino acid is amino acid that the body can produce on its own. Example of the non-essential, because something that is very cheap is not important. If my body can produce it, then that means it is cheap. But if I'm buying it with money from outside, then it is essential. So essential amino acid is like petrol. Now a gallon of petrol is around 11 cedi. It is essential. When it was around 5 cedi, it was not essential because we're not buying at high rate like that. Conditionally non-essential means that the body can produce it when it is needed. So the body can go through several reactions to produce it as and when it is needed. But if it is not needed, the body will not produce it. So that is conditionally non-essential amino acid. Do you understand? Okay, so you go and look at how tyrosine is synthesized from phenylalanine. When you read the footnotes or the legend of this figure, you will understand. Okay, uncommon amino acids. Some amino acids, uncommon amino acids also have important functions. In addition to the 20 common amino acids, proteins may contain residues created by modification of common residues already incorporated into a polypeptide. So post-synthetic modification. This means that some amino acids can be modified. So let's see, by putting some elements or atoms there. So let's see, 4-hydroxyproline is found in collagen. 5-hydroxylysine found in collagen. 6-amino or 6N-metallysine, constituent of myosin. Myosin is what? Protein found in what? Muscles. Gamma-carboxyglutamate is a prothrombin. Prothrombin mediates what? Blood clotting cascade. then selenocysteine. Can you see the selenocysteine? Hello. Okay. So, uncommon amino acid structures. So these are some of the uncommon. They said the meeting will end in 5 minutes time, so I will resume again. So I will pause the video when it is the recording when it is done. So we have some uncommon amino acids structures. Proline. This is proline. When you add OH over here, it becomes hydroxyproline. Lysine. This is lysine. When we add OH over here, it becomes hydroxylysine. You know, OH is called hydroxy group. Okay, tyrosine. When we add I'm coming. Can you see what is down there? Okay, so when the thing has come again. So this is tyrosine. When we add OH and another aromatic group there like this, it becomes what? Tyro. It becomes tyro. It becomes tyrosine. The S will change to X and H will come and add. Why are you studying all this? Let's continue. So these are uncommon amino acids. So we have the gamma carboxyglutamate, selenocysteine. We add a lot of different atoms, molecules, and making this structure a homogeneous structure. And they are all found in nature. So transient modification of amino acids. We can add a phosphate group to an amino acid through the process of what phosphorylation. When we add a methyl group, let's see the structure of a methyl group. So this is a methyl group, CH3. It's methyl group. Phosphate group is PO4, 2 minus phosphate. Can you see? Hello?
No, sir.
You can see?
Yes, please.
You can see this thing, the whiteboard.
Yes, your picture is on it.
You said?
Okay, sir, we've seen it.
Can you see?
Yes,
This
sir.
is a methyl group. And this is a phosphate group. Where the pen is pointing. This is a phosphate group. Can you see?
Minus.
And this is what? A methyl group. It's not CH3 minus, so it's CH3,
CH3.
Okay.
Yes, sir.
Okay, so let's go back to the main lecture. So, acetyl group. Acetyl. That's acetate. Then ADP ribosylation. You see the structures. Let's go. So this is aspartic acid. When we remove this COO, this carboxylate, or yes, when we remove this group, we get what? And put a phosphate group here. Aspartic now changes to phospho. It changes to what? Phosphoserine. Phosphoserine. So this amino acid is now called phosphoserine. It used to be called aspartic acid. Now it is called what? Phosphoserine. We just phosphorylated. We remove the COO and replace it with phosphate group, giving us So these are examples of phosphorylated amino acid: phosphoserine, phosphothreonine. We have just introduced phosphate group, phosphotyrosine. Okay, so you can go and learn inborn errors of metabolism of amino acids: phenylketonuria, alketonuria, albinism, and the rest. Hello? 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 Okay, so peptides are changed. Please mute your mics. Mute your mics. Peptides are chains of amino acids. So we are now on peptides. Two or more amino acid molecules are joined through a peptide bond. Such a linkage is formed by... Oh, please, mute your mics. Felicia, mute your mic. Okay. Two or more amino acid molecules are joined through a peptide bond. Such a linkage is formed by removal of water, that's dehydration, from the carboxyl group of one amino acid and the amino group of another amino acid. Okay, so let's see how a peptide bond is formed. So amino acid, a bond between two amino acids is called what? Annabelle. A bond between two amino acids is called what?
Bond between two amino acids. Peptide.
Okay, it's a peptide bond. So the bond is between the amino group over here and the carboxyl group over here. So the carboxyl group will donate OH and the amino group will donate H. So the H and OH will react to form what? Water. So this is it. The H and OH will react to form this water. So when they react to form the water, they will leave a bond here like this. Do you understand?
Yes, sir.
Okay. So, hydrolysis reaction of amino acids. Hydrolysis is what? The first one we did was dehydration or condensation reaction with the removal of water. This one is rather, hydrolysis is what? We are breaking two, this is a bond already. The first one was not a bond. The first one we were trying to establish a bond. But the second one here we are trying to what? Hydrolyze or use water to break this bond. So when we add water to this bond, it will break it. So now this CO, this O will go and join to this C double bond O to form this O minus. Then the H2, one H will come and join, 2H will come and join to the NH to form what? NH3. So we have restored the two individual amino acids back to normal way of life. So sequence of amino acids, we have dipeptide, oligopeptide, tripeptide, polypeptide, tetrapeptide, and protein. What is a dipeptide? Felicia. Please, your mics. More than two people have opened their mics. So when you open your mic, just talk fast. Felicia, what is a dipeptide? No. Two amino acids. It's what? A peptide containing two amino acids. Tripeptide contains what? Felicia. Tetrapeptide contains what? Eh? Eh? Tetrapeptide. Eh? Felicia, hurry up, we are recording. Hurry up. Tetra, four amino acids. Oligopeptide contains, Oligopeptide contains more than 3 amino acids, but between 3 to 10 amino acids, that's oligopeptide. Polypeptide contains more than 10 amino acids. Then protein contains more than 10,000 amino acids. So let's look at the dipeptide. I said dipeptide contains what? 2 amino acids. So this is a dipeptide bond between 2 amino acids. Amino acid 1, amino acid 2, forming a dipeptide bond. Tripeptide contains what? 1 amino acid, 1 amino acid, 1 amino acid. So how many amino acids are here? How many amino acids are here? So how many peptide bonds are here? How many peptide bonds?
3 peptide bonds.
You are lying. This is valine. This is threonine. This is cysteine. So the bond between valine and threonine is one bond. This one, where the arrow is. Then the bond between threonine and cysteine is this one. And so how many bonds are there?
2.
So 2 peptide bonds. 2 peptide bonds. Okay, so let's look at it. So this is it. Let's look at another one. This is tetrapeptide. How many amino acids? 4. Mention them. Hurry up, I'm recording. Mention them. Their names are written there. Clap for yourselves. Hey, Munimadio. So, if I were to be like you by this time, my life would have changed. So let's continue. Mute your mics and let's continue. So, how many peptide bonds are here?
73.
Very good. So let's proceed. This one, this one, this one. Okay. Okay, so tetrapeptide. Okay. This is a oligopeptide between 3 and 10 amino acids. This is a polypeptide. You can see the polypeptide has about greater than 10,000. Plenty amino acids. Okay. Then this is what? No, polypeptide has plenty amino acids, but it's less than 10,000. It's less than 10,000. Then a protein has greater than 10,000 amino acids. So you can see you cannot even count it. So this is a table. Go and study it. Cytochrome C is in the liver. It is the number of polypeptides chain is 1. Apolipoprotein is what? A type of what? Lipid. 1. Titin, so chymotrypsin, they are all enzymes. Go and study them. Number of chains, number of residues. Number of residue means what? Number of what? Amino acids residues. Okay, so how do we name a peptide? Annabelle, how do you name a peptide? Okay, by
Convention.
a year, that's it. By convention. Pentecost convention. Let's continue. By convention, the free amino acid end, the end terminal of the peptide chain, is written to the left, and the free carboxyl end C terminal to the right. Therefore, all amino acids are read from the N terminal. N terminal means the amino side to the C terminal, which is the carboxyl side. So let's continue. So how do we name peptides? We have the N terminal end. This is the N terminal, the amino side. And this is the C terminal, the carboxyl side. So when a peptide is named, all amino acid residues have their suffix. The suffix is the starting words, and the ending words. Ein, an, ic, or ik, or et, change to I, with the exception of carbon, the carboxyl terminal amino acid. You will understand very soon. So, meaning that aspartic acid will change to what? Aspartar. Glutamic acid will change to glutamal. Arginine will change to arginyl. Lysine will change to lysyl. Histidine will change to histidyl. Asparagine will change to asparaginyl. Glutamine will change to glutaminyl. Serine will change to serial. Threonine will change to threonal. Proline will change to prolyl. Isoleucine will change to isoleucyl. Do you follow the trend?
Yes, sir.
Okay, so let's go. So the amino acid at the N terminal, this area is the N terminal, and this is the C terminal. Meaning that this is this side is the amino side, and this is the carboxyl side. So this amino acid at the left hand side will be named as what? Valyl. So valyl, but this one, the alanine will not change. So this one becomes valyl alanine. So let's name it. So valyl alanine. Do you understand?
No, sir.
Okay, let's go to the whiteboard. What I mean is that this is an amino acid, H, C, R chain. This is the carboxyl side. This is the amino side. So assuming this is amino acid A, and it is called, let's change it to what? Glycine. Okay, amino acid B is here. Amino acid B is here. Let's draw it. COOH NH2 is constant, COOH is constant, H here is constant. The only change is here, the side chain, so CH3. This is what? Alanine. Alanine. Can you see the alanine?
Yes, sir.
And this is, the first one is called glycine. So if they are joined together through a peptide bond, then how do you name it when there is a bond between glycine and alanine? Because the glycine is at the left hand side, so we assume that the left hand side is the amino side and the right hand side is what? The carboxyl side. Do you understand?
Yes, sir.
So the amino side, when you name it, you add Y glycol. And the one at the right hand side, which is the carboxyl side, no, you will leave the name like that. Alanine. Do you understand?
Yes, sir.
Okay, so let's go back. So this is threonine, glycine, and tyrosine. So how do we name it? Annabelle.
Sir, please. Alanine.
Hey, Samu. There is threonine, glycine, and tyrosine. Where is the alanine? There's no alanine here. Come again.
Please, which one?
What? Are you not seeing threonine, glycine, and tyrosine?
Except please, I'm seeing it.
And so how do you name it?
So this one is true now.
And
the glycine becomes glycol.
Uh-huh.
And the tyrosine becomes pyrosol.
And yet tyrosine. The tyrosine is at the right hand side. That's the last end. So it will remain the same name. It is at the carboxyl side, so it will remain the same.
Okay, so it becomes tyrosol.
Uh-huh.
Glycine now, tyrosine.
Very good. So let's see. So it becomes what? Threonine, glycol, tyrosine. Can you see? Can you see?
Yes, sir. Yes, sir.
Okay. So Felicia, name this one for me. Felicia, don't waste time. Oh, time is going. The recording will be over. So hurry up. Okay. Hey, Felicia is not talking. Felicia. Eh? Phenylalanine, methionine. Very good. So let's see it. So can you? But you can see now. Can you see? Can you see? Can you see?
Yes, sir.
Okay. So let's go on. So the same procedure for the rest, eh? So this one too. Glutamal, lysyl, valyl histidine, eh? Very good. Okay, so let's look at protein structure. Later I'll be doing it, but let's just take a glance through what a protein is. Protein is what? We have the primary structure. The primary structure is a single chain. Let's see. So primary structure. Then the secondary structure, we have the alpha helix, beta plate, and beta turns. We have the tertiary structure and the quaternary structure. So let's see. Whenever you see a long chain of amino acid, it is called primary structure. When the long chain of amino acid, there is a bond between the turns. There is a bond between what? The helix. This is a helix. beta pleated sheets, alpha helix. This is an alpha helical structure. So there's a bonding, van der Waals forces and the rest between them. We call it what? Secondary structure. When this protein is, this polypeptide is folded, this same protein is folded like this, it becomes what? A tertiary structure. When the tertiary structure, we have about more than one put together, it becomes what? A quaternary structure. So this one has what? Four polypeptide, folded polypeptide put together. So it becomes what? Quaternary structure. So this is a hemoglobin chain. It has the alpha globin or alpha polypeptide and beta globin. So let me illustrate it on the whiteboard. So are you with me? Are you with me? Okay. We'll finish very soon. So let's see. Let's see now. So if we have can you see? Hello? Hello?
Sir.
Are you with me?
Yes, sir.
Okay. Hey, the whiteboard is not.
Yes, sir.
Okay. So if you have this, with one amino acid, one amino acid, one amino acid, one amino acid, one amino. How many amino acids do we have?
5.
And how many peptide bonds?
4.
Very good. Felicia, are you following?
Yes, sir.
Raphael, are you following?
Yes, sir.
Okay. So let me save this recording so that or else it will not be able to load for you.